Welcome to the lecture series in food microbiology. This is the fourth lecture in the series of foodborne infections. So far we discussed about the infections caused by Listeria, Salmonella, Shigella, and in the last lecture we discussed about the Vibrio species, especially the most important counterpart, the Vibrio cholerae, and we discussed about some of the diseases which are caused by enteric viruses. The predominant one or the most commonly heard one would be the jaundice, which is caused by hepatitis A, which is a foodborne infection. But along with that, you have other foodborne viruses also, like your Norwalk-like viruses. In this lecture, we are going to discuss about the foodborne infections, which is caused by the common gut microbiome E. coli. E. coli, as you know, or Escherichia coli, is a bacterium which is prevalent everywhere. And this is a common intestinal dweller. All mammalia contain E. coli in their gut. And normally, this is not a pathogen. We call it as an opportunistic pathogen. This is a part of your gut microbiome. E. coli is an almost universal inhabitant of the gut of humans and the other warm-blooded animals, where it is the predominant facultative anaerobe, though only a minor component of the total microflora. So you have so many organisms which are present in your gut microbiome. E. coli is one among them, and it being there normally does not implicate any illnesses. Among so many millions of microbes which occur, this is among the part, that is all. Generally a harmless commensal, it can be an opportunistic pathogen causing a number of infections like gram-negative sepsis, urinary tract infections, pneumonia in immunosuppressed patients, and meningitis in neonates. So, it normally is a commensal organism, it is harmless. But given an opportunity, when you are immune compromised, it can be the causative reason for sepsis, urinary tract infections, pneumonia, and meningitis. Its common occurrence in fecus, ready culturability, generally non-pathogenic character, and survival characteristics in water has led to the adaptation of E. coli as an indicator of fecal contamination, and as such the possible presence of enteric pathogens like Salmonella typhi in water. As we saw in the earlier lectures, we talked about indicator organisms whose presence beyond a particular number can tell that this particular water is not fit to be consumed. We talked about water potability, and we discussed about what we call as an IMVIC test. There we said that we look for E. coli, gram-negative enteric pathogens. So, if E. coli is present beyond a particular number, it means that there is some contamination with sewage somewhere in that particular water and that it is not fit to be consumed. So, this is taken as an indicator organism, especially a fecal contamination. The strains of E. coli were first recognized as the cause of gastroenteritis by workers in England investigating summer diarrhea in infants in the early 1940s. The E. coli which causes gastroenteritis has been divided into four major groups: the enteropathogenic E. coli called as EPEC, enteroinvasive E. coli called as EIEC, enterotoxigenic E. coli called as ETEC, and enterohemorrhagenic E. coli called as EHEC. This is based on the type of the illnesses and the changes they cause inside the GI tract. Recently, they have been subdivided into 6 groups. So, the other 4 plus 2 recent ones, E. coli called as EGGEC, which is called as the enteroaggregative E. coli, and then diffuse adhering E. coli called as DAEC. These 2 ones have now been added to the already established 4 types which cause gastrointestinal diseases. Enteropathogenic E. coli or EPC. These strains do not produce enterotoxin, but they do cause diarrhea. These exhibit localized adherence to tissue culture cells and agglutinate in tissue culture medium. So, if you isolate these E. coli enteropathogenic strains and inoculate them in cells, intestinal cells which have been cultured in the tissue culture medium. They adhere to the tissue culture cells and agglutinate them, and they possess the adherence factor plasmids that enable adherence to the intestinal mucosa, which means that they have certain plasmids which produce certain factors. These factors are called as adherent factors. These adherent factors would help these organisms to go and stick on to the intestinal mucosa, mucosal cell lines, the epithelium of the mucosa. After colonizing the intestinal mucosa, the attachment effacement or called as lesions are produced. So, once they attach themselves to the mucosal cell lining, then they start producing lesions on the lining. The process starts upon initial contact and is helped to be aided by a plasmid encoded bundle forming pilus. Upon reaching the gut by food, the enteropathogenic E. coli first gets into contact with the intestinal cells, and this contact is helped by the formation of a bundle forming pilus. The gene for this formation of this pilus is plasmid encoded. Then you have the EPEC secreted proteins, which is called as the ESPS. They block phagocytosis. See, normally phagocytosis is something which the body's immune system does to kill what it identifies as pathogens. So, phagocytosis is resisted here because of the help of these proteins which are secreted by the organism, and this leads to the skeletal rearrangement and tyrosine phosphorylation of TIR. So, when the TIR binds with the outer membrane protein intimin, the attachment is intimate, resulting in the destruction of the brush border microvilli. It escapes phagocytosis following with cytoskeletal rearrangement, autophosphorylation of the tyrosine phosphorylation. Receptor tyrosine kinase normally is something which is present. It is a transmembrane protein which acts as not only a channel but also as a signal receptor. It normally two of the receptor tyrosine kinases autophosphorylate when brought together in response to a signal which is received. So, you have a tyrosine phosphorylation there which causes the binding with the outer membrane protein intimin, which is a part of the cytoskeletal system. Because of all this, the attachment becomes fixed. This results in the destruction of the brush border microvilli. EPEC strains generally cause diarrhea in children under the age of 1, and these strains are important in infant diarrhea worldwide, especially in places with poor sanitation. They are transmitted directly or indirectly through human carriers. Several serotypes of EPEC are implicated in waterborne and foodborne disease outbreaks in different countries. So, EPEC strains normally are very common in underdeveloped countries where malnourishment is very common and proper nutrition is not available for children, especially below the age of 1. Sanitation procedures in these countries will also not be proper. Hence, the spread of this children suffering from this would be very common there. And you have several serotypes which are implicated in both as waterborne and foodborne disease outbreaks in different countries. This is how once the pathogen has entered the gut, it goes and bites again the brush border microvilli. This is the intestinal epithelium, intestinal microvilli epithelium. This goes and settles there, attaches itself, causing the diarrhea. This binds there, causing the outflow of water from the intestinal lumen to the outside, causing what is called as unabsorbed nutrients and water, which are expelled as the diarrhea here. Enterotoxigenic E. coli, which is called as ETEC, otherwise called as travelers diarrhea. This is the major cause of diarrhea in travelers as well as infants, especially in developing countries with poor sanitation. The diarrhea or the illness caused by ETEC strain usually occurs between 12 hours and 36 hours after ingestion of the organism. The symptoms can range from mild febrile diarrhea to severe cholera-like syndrome with watery stools without blood or mucus, stomach pains and vomiting. The illness is usually self-limiting, persisting for 2 to 3 days. The ingested organism results expulsion from the small intestine with the rapidly flowing chyme by adhering to the epithelium through attachment or colonization factors in the form of fimbriae on the bacterial cell surface. Once you have consumed a food containing ETEC, this goes into the gut, and in the small intestine, instead of being expelled out with the fecal matter, it resists the flow by attaching itself to the epithelium with the help of colonization factors in the form of fimbriae, and these fimbriae are present on the bacterial cell surface. Two toxin types are produced: the heat stable toxins, which are called as ST. The heat stable toxins can resist heat to 100 degrees centigrade for 15 minutes. They are acid resistant. The other type of toxins are the heat labile toxins, LT. These are inactivated at 60 degrees centigrade after 30 minutes and at a low pH. So, ETEC strains can produce two types of toxins: heat stable toxins and the heat sensitive or the heat labile toxins. The heat stable toxins are so stable that they cannot be destroyed even at keeping at a temperature of 100 degrees centigrade for 15 minutes together, and they are resistant in an acid resistant, meaning acidic environment will not destroy them. The heat labile toxin, which are inactivated at 60 degrees. So, till 60 they can withstand the heat, but making it stand at 60 for more than 30 minutes, the heat labile toxin will be destroyed and they are inactivated at low pH. The LT1 bears a strong similarity to the cholera toxin, that is, the heat labile toxin of ETEC is similar to the cholera toxin. It has 5 subunits which are responsible for binding of the toxin to the epithelial cells, and the A subunit which is translocated into the epithelial cell, where it activates the adenylate cyclase, just like what the cholera toxin does. The activation of adenylate cyclase in turn increase in the CAMP levels, then inhibits the sodium and chlorine and water absorption by the villi cells and stimulates their loss from the intestinal crypt cells, thus leading to watery diarrhea. Two types of heat stable toxins have been recognized. The most common, STA, this is a low molecular weight, poorly antigenic polypeptide of less than 20 amino acids produced from a 72 amino acid precursor. So, the protein, heat stable protein or the toxin is less than 20 amino acid long. The precursor molecule is a 72 amino acid molecule long. It is a poorly antigenic polypeptide. Its resistance to heat, low pH and proteolytic digestion probably derive from its compact 3-dimensional structure, which contains 3 disulfide linkage. This heat stable protein A acts by stimulating the production of cGMP by guanylate cyclase in the epithelial cells. The STA differs from the LT, which is the heat labile toxin, in that the heat labile toxin stimulates adenylate cyclase, whereas the heat stable toxin stimulates guanylate cyclase. And similar to the heat labile toxin, your cholera toxin is also stimulating adenylate cyclase. STB, the other type of heat stable toxin, elevates the luminal 5-hydroxytryptamine and the prostaglandin E2 receptor, both of which are mediators in intestinal secretions. When these are stimulated, that in turn will lead to the symptoms of diarrhea. The pathogen is spread directly or indirectly by human carriers. Both food and water have been implicated in the outbreak, and sporadic cases in humans. An ingestion of a large number of cells, 10 to the power of 8 to 9 cells, is necessary for the individual to develop symptoms. This is a picture of the ETEC enterotoxigenic strain. The organism comes here, gets attached to the villi, causing the change, in turn causing the reaction in the intestinal cells, stimulates cGMP here. The stimulation of the cGMP in turn would allow open up the transmembrane channel, leading to the secretion of the ions and water, leading to the symptom of diarrhea. Entero-invasive E. coli, otherwise called as EIEC. These strains are known to cause dysentery similar to that is caused in shigellosis. The ability of the strain to produce an invasive factor is thought to be the cause of the disease. Human carriers are the reason for the spread of the disease. It's either direct or indirect. Then you have the enterohemerogenic E. coli. The strains in this group, the principal zero group is 0157:H7, has been recognized relatively recently as the cause of bloody diarrhea. This is called as hemerogenic colitis and hemorrhagic uremic syndrome in human beings. So, enterohemerogenic E. coli, the well-known example in this category is 0157:H7, causes hemorrhagic colitis and hemorrhagic uremic syndrome in humans. The primary carriers are cattle. Ingestion of a few as to 10 to 100 cells can produce the disease, especially in sensitive individuals. 3 enterotoxins, which are called as the virotoxins, are produced by the serotype which causes the disease symptoms. Let us discuss the toxins which are produced by both EIEC, the enteroinvasive E. coli, and the 0157. The pathogens produce several polypeptides, the genes of which are included in the plasmid. These plasmid encoded genes which produce polypeptides, these are considered to be the invasive factors that help the pathogen to invade the epithelial cells and set up infection in the colon. But separate toxins as such now have not been identified for EIEC. EIEC E. coli, only humans are known to be the host of the pathogen, and the food can get contaminated either directly or indirectly through fecal contamination. The EIEC pathogens are sensitive to pasteurization temperature. So, proper heat treatment, elimination of post-heat contamination for a ready-to-eat food, and refrigeration of the food soon after preparation. All the 3 things are required to control the disease. In addition, proper sanitation at all stages of food processing and handling is an important factor. Gastroenteritis due to EHEC, the enterohemerogenic E. coli. The primary or the principal serotype associated with this is E. coli 0157:H7. This produces a virotoxin VT1. This is also called as a sugar toxin ST. So, the strain is designated as either VTEC or STEC, and more than one toxin has been implicated in the disease and the symptoms related to it. In humans, the EHEC colonizes the large intestine. Once it colonizes the large intestine, the toxin released by EHEC binds to the endothelial cells expressing GB3, allowing absorption into the bloodstream and dissemination of the toxin to other organs. The tissues and the cell types expressing the GB3 varies among the hosts. So, the distribution of the GB3 targets the pathology of the toxin mediated disease to the cells expressing GB3. So, whichever are the cells which show or which have a larger number of the GB3 receptor, those are the cells which are going to be bound by the virotoxin. For example, the renal glomerular endothelium expresses high levels of GB3 in humans. So, sugar toxin production results in acute renal failure in this case, thrombocytopenia and microangiopathic hemolytic anemia. So, the cells or the tissues which have the GB3 receptor are more prone to sugar toxin than the other tissues where the distribution of GB3 receptor is not much. So, this is how the pathology works out in the case of an enterohemerogenic E. coli. The receptor is bound and the organ is bound, and you have the symptoms causing the manifestation in the tissues concerned. Enteroaggregative E. coli. EAEC, or otherwise called as EAGGEC, it is a pathotype of E. coli which causes acute and chronic diarrhea in both developed and the developing world. This also causes urinary tract infections. The pathogenesis of EAEC involves the aggregation of and adherence of the bacteria in the intestinal mucosa, where they may elaborate the enterotoxins and cytotoxins, damage the host cells, and induce inflammation that results in diarrhea. EAEC is a pathotype which can cause urinary tract infections also. So, these pathotype aggregate and then adhere to the intestinal mucosa, and once adhere to the mucosa, they secrete the enterotoxin and the cytotoxin, damage the host cell and induce inflammation. The diagnosis of this pathotype is problematic. Serotyping alone cannot identify the strains of E. coli expressing the EAEC phenotype. Currently, the detection or identification of EAEC is either by cell adhesion test or DNA based tests involving gene probes. So, you identify a specific gene which is peculiar to the EAEC type, synthesize a primer and then use that primer to probe in a polymerase chain reaction to find out whether the infection is by the EAEC strain for detecting the genes in the encoding the cell adhesion. The genes which are used as a primer in the polymerase chain reaction would be for the genes which are coding for cell adhesion in the pathotype. The other recent discovery pathotype was the diffuse adhering E. coli. It is defined by the pattern of diffuse adherence in which the bacteria uniformly cover the entire cell surface. So, instead of sticking on to a particular receptor which it prefers, the DAEC E. coli spreads throughout the cell surface, and this is supposed to cause diarrhea in malnourished children. Not much is known about these two types, pathotypes, DAEC as well as EAEC, were recent additions to the E. coli pathotypes which cause gastrointestinal diseases. Detection. Diagnostic Automation Inc. company offers an E. coli virotoxin food ELISA kit. That is an enzyme linked immunosorbent assay to screen food products for the presence of virotoxin. This virotoxin food ELISA kit is a quick, economical and sensitive food toxin kit which is designated to detect the virotoxin antigen in the food. So, your EHEZ strains which synthesizes these virotoxins and the presence of the toxins in the food can be detected by this ELISA kit. The mechanism of the Shiga toxin, the STX1, STX2, STX2E and the castobrine protein resin are the same in the sense that these are commonalities. Remember that for the strains of the EHEC, we said that it is either the Shiga toxin or the EC. So, the mode of action of these toxins are similar. These are N-glycosidases that cleave a specific adenine residue from the 28th subunit of the eukaryotic ribosomal RNA, leading to the inhibition of protein synthesis. The main complication of the enteritis is dehydration due to diarrhea. Any enteric disease, because of diarrhea and vomiting, the major problem caused is by dehydration. So, drinking fluid and keeping up with the hydration is very important in any enteric disease. If the liquids cannot be kept down due to the intense vomiting or diarrhea, then intravenous fluid therapy might be required. Antidiarrheal medications are sold over the counter at drugstores. Normally, for many of the enteric diseases, antibiotics are normally not recommended unless it is dire. Summary. Any foodborne infection is caused by several enteric pathogenic bacteria as well as viruses. The viable cells and the particles survive in the GI tract or the GI environment and set up an infection in the intestine to produce the enteric symptoms and fever. This is common to all enteric diseases, whether it is a bacterial disease or a viral disease. Once the cells reach the GI tract, they survive in the GI tract and set up an infection in the intestine to produce the enteric symptoms as well as fever. Several can also infect the organs inside the body and produce specific diseases, like we saw in the case of hepatitis A. Once it reaches the gut, it migrates to the liver and there it shows the symptoms. So, not only in the enteric area, but it can also cause disease in the cells to which they migrate. And several pathogens of this group can also produce high rate of mortality. All of them are sensitive to heat, low heat, and are killed by pasteurization. Proper sanitation, heat treatment, and refrigeration can be adopted to reduce their incidence. However, a few can multiply even at 1 degree. To overcome the problems with them, food should not be refrigerated for long, and the refrigerated food should be reheated uniformly before consumption. So, in general, any enteric organism, whether it is going to cause disease in the intestinal area or whether it migrates to other cells, is going to produce a diarrhea and vomiting as a symptom along with fever. And most of them either produce a toxin or react with the intestinal cells, causing the symptoms. Most of them can be destroyed by heat. So, well cooked and well boiled water or safe water drinking and proper sanitation would be the best precautions for avoiding the incidence of such diseases. So, for a bacterial disease or for a foodborne viral disease, what would be the common precautions you would take, especially if you are traveling to an area which is endemic to a disease? This would be the question we would pose after seeing the sessions on foodborne infections. Thank you.